This is Blood and Cancer, the official podcast of MDedge Hematology Oncology. I'm the voice of MDedge Podcasts, Nick Andrews. And welcome to this, in case you missed it, rehash of one of our very best and most important episodes that Blood and Cancer has done. This episode was originally published back in February and features Dr. Gary H. Lyman in a discussion all about biosimilars, including how they're made, which ones were approved as of February 2019, and extrapolation and interchangeability, as well as where some biosimilars stand in both ASCO and the NCCN guidelines for patients who are going into supportive care. Just a quick reminder, if you haven't already, please take a moment to complete our short listener survey. We really appreciate your feedback, and of course, the survey is anonymous. Also, a quick note, Dr. David Henry and myself will be at the 2019 American Society of Hematology meeting in Orlando next month to record new interviews for Blood and Cancer. So if you're going to be there, email us or tweet at us and plan on coming by and saying hi. Okay, it's time for This Week in Heme Onc. This week in Hematology Oncology, we have a bit of an ASH preview for you. The key themes this year at the meeting will include tackling CAR-T obstacles, sickle cell advances, and VTE. New findings regarding CAR-T cells are taking aim at the drawbacks of therapy, such as the time, expense, and toxicity that are involved. This is according to Dr. Robert A. Brodsky, who is the ASH Secretary and Director of the Division of Hematology at Johns Hopkins University in Baltimore. As an example, he noted a study slated for presentation during a plenary session at the upcoming annual meeting that involves the investigational T-cell bispecific antibody mosunetuzumab, which targets both CD20 on the surface of the malignant B cells and CD3 on cytotoxic T cells, engaging the T cells and directing their cytotoxicity against B cells. In a study which will be presented at the meeting as abstract number 6, 218 non-Hodgkin lymphoma patients, including 23 who had already received CAR T-cell therapy and had relapsed or who were refractory to the treatment, 64% responded and 42% had a complete response, and the median duration of response is about 9 months. He added that the preliminary results suggest that it is less toxic than CAR T-cell therapy. Also coming up at the meeting, there are 2 CAR T-cell therapy-related studies that address the therapy's application for multiple myeloma. The 2019 annual meeting is taking place beginning December 7th. You can stay up to date on all of the clinically relevant news and data from the meeting at mdedge.com/hematology-oncology. Coming up next, our In Case You Missed It episode with Dr. Gary Lyman, all about biosimilars. Welcome back to Blood and Cancer. I am Nick Andrews. Okay, it's time now for our interview from February 2019 with Dr. Gary Lyman and Dr. David Henry.
I'm Dr. David Henry, and I'm delighted to be talking today to Dr. Gary Lyman, who's in Seattle, and I'll introduce him first with his title, and then we'll have our conversation, which tonight is going to be about biosimilars. So Gary is a member of the Public Health Sciences and Clinical Research Divisions at the Fred Hutchinson Cancer Research Center in Seattle. He serves there as the Senior Lead for Healthcare Quality and Policy in the Hutchinson Institute for Cancer Outcomes Research. He's also a Professor of Medicine at the University of Washington School of Medicine, Adjunct Professor in the University School of Public Health and Pharmacy. He's Executive Officer of SWOG, SWOG Cancer Research Network. Dr. Lyman chairs the ASCO Task Force on the role of observational research in clinical practice and the ASCO Working Group on Biosimilars in Oncology. He's a member of the Society's Value of Cancer Task Force, Cancer Care Task Force, and Cancer Research Committee. He also finds a little bit of time to serve as editor-in-chief of the journal Cancer Investigation, is on the editorial board of several specialty journals. And as wonderful as that introduction is, he is the most modest, hardworking man I know. So Gary, welcome to this podcast.
Thank you very much, David. It's a pleasure to be with you and discuss this topic. It's always good to talk to you.
Well, likewise, and thank you. And then we begin by talking about what's a biosimilar. You know, when I first heard this, and actually heard you and Jeff Crawford speak on it, it's a shame it's called biosimilar because your brain hears similar, yet really that's not the case. So maybe if you could begin by defining, as it comes to our shores and into oncology, what's a biosimilar?
Sure. So the FDA in the U.S. has formally defined a biosimilar as a biologic product that's highly similar to the reference product, so the original biologic treatment, notwithstanding what they call minor differences in clinically inactive components. In the end, they conclude an agent is a biosimilar if it has no clinically meaningful differences with the original biologic product. And in terms of safety, in terms of efficacy, and then in terms of purity and potency, based largely on preclinical data. So in the end, as you know, these are biologic substances. They're produced in living systems, and therefore they cannot be replicated identically because of the complexity of their production, their isolation and packaging. So what the FDA's goal here is, as the patents expire on the original biologics, to encourage companies to come forward, because there's money to be made, by producing a product, a biologic substance that's produced in a very similar fashion to the original product, and to demonstrate that it's highly similar, largely based on preclinical information. So it's molecular structure, it's preclinical pharmacokinetics in animal systems, and then in early human studies, it's PK, PD, looking for any antibodies, immunogenicity, and then only after that, if needed, provide additional clinical safety and efficacy data. In the end, their focus is much less on large randomized controlled clinical trials that the originator had to go through, because if you required that of all subsequent biosimilars, of course, the cost of production would also be extremely high, and you wouldn't have an incentive to bring down the price of these expensive drugs, which is one of the goals of this whole process, to lower costs and enhance access. Because
so if I restate it, yeah, go ahead.
Go ahead. Well, I was just going to add, because of the way this is done, the FDA has one additional provision, which is, and that we'll get into, is the extrapolation. That is, if the original drug was approved based on data in several different indications, and a biosimilar comes along and demonstrates safety and efficacy for one of those, the FDA has the authority to grant extrapolation to these other indications without requiring additional data. However, they may impose, and probably will impose, fairly extensive post-marketing surveillance in case some delayed, unusual, rare side effect comes along.
So to restate it, from my understanding, if I go down the aisle at CVS and looking for aspirin, I can find a generic. It's a chemical. It's a simple chemical. They should be identical. But in this biosimilar issue, especially in oncology, these are very complicated molecules, very large molecules. They're made not in a factory built from the ground up, but made in nature's factories, cells, and there's different kinds of cells. In fact, one biosimilar may be made in a different cell from the brand originator product. And so they're much more complicated and never exactly identical. In fact, I always think of rituximab, which comes out in 1995, is likely a biosimilar of itself due to change in manufacturing and process over time, so-called drift. So, in fact, we use probably biosimilars every day, whether or not we know it.
Absolutely, and that's an important message that I think many of us had not been fully aware of until this whole stream of biosimilars began to come along, and that is the rituximab that was approved back then based on data provided to the FDA is not exactly the same as the rituximab you pull off the pharmacy shelf and give to patients today because of this drift, just minor changes in production, in components, and just the mix of time will have changed in subtle ways these agents. So the charge of the FDA is to prove over time that none of these changes, which are unavoidable, have a meaningful impact on safety and efficacy for patients.
So we take, for example, let's take trastuzumab, which has a biosimilar out, I know approved. I don't know how much on the market is out or usage, but what did they have to go through to get that through the FDA committee and approval process? As you mentioned, if they did the whole thing, it'd be prohibitive expensive wise. So what studies did they have to do to say, okay, here it is, and the FDA says fine?
Yes. So this has, of course, been a process in evolution at the FDA. So the good news is we have data from Europe going back a decade using very similar review and approval processes that have led to approval of many biosimilars in the EU, and none of these have had to be removed because of safety and efficacy concerns after they've gone through the EMA approval process. But it took longer for the FDA to get the rules in place. And the first biosimilars began to come out a couple of years ago in the supportive care arena. We're now, as you point out, in an era where we have some cancer treatments, biologic cancer treatment biosimilars approved, specifically trastuzumab and bevacizumab. These have, to my understanding, have not hit the shelves yet, but and there may be some patent issues that are still being litigated in some cases. But these will be coming very shortly, along with a whole slew of others. And the approval of these is based, as I indicated earlier, largely on preclinical data or preclinical trial data. So first of all, they have to demonstrate that it is the same molecule, or at least the same amino acid sequence, similar structure, that it, again, in animals and then in humans, the pharmacokinetics and pharmacodynamics are very similar. And very critically, they need to demonstrate that there's no greater incidence of antibody formation, immunogenicity, than there is to exposure to the original product. So specifically a concern around neutralizing antibodies that might reduce or prevent the effective use of these treatments in cancer treatment, or lead to allergic responses. So again, the focus is on doing all this molecular characterization using a whole host of tests on the molecular details of the molecule, that it's as close as one can get to the same agent. Do the preclinical studies in animals, and then often in healthy humans, just make sure that the pharmacology is essentially the same as the original product.
And to make us prescribers feel good, they each, for example, trastuzumab and bevacizumab, each has done at least a large phase 3 or somewhat sizable phase 3 trial against brand product and shown similar safety and efficacy?
Yes. Usually these are not what we have thought of historically as large phase 3 comparative trials. They tend to be on the smaller side, but they are comparative trials looking at, again, some of these safety and efficacy endpoints, but generally just one. And so instead of the historical experience of, requiring 2 or 3 large phase 3 randomized trials, there's much less emphasis and requirement for the large numbers of patients being treated in these randomized trials. So the evidence supporting them from the clinical comparative standpoint is less. But as I said, the reason for that is you've gone through all these other hoops. We have a great deal of experience from Europe. We have experience with the originator product. So the FDA is convinced, and many of us working in the space are convinced, that this is a very rigorous process that does due diligence to demonstrating the efficacy and safety of these agents. We have to keep our eyes and ears open if something shows up and post-marketing surveillance will be important, but the bulk of the evidence we have is that these can be safely used in the clinic.
So if we leave this portion of our discussion with just naming a few, so I think we've covered trastuzumab, bevacizumab, and I think in supportive care we have an erythropoietin biosimilar and a couple of G-CSF biosimilars.
Yes, we do, and of course they keep changing almost daily. There are now 14 biosimilars approved by the FDA. As you point out, at least the first ones and the majority are in the supportive care arena. The Zarzio or filgrastim SNDZ, and this gets into the nomenclature issue, which is quite bothersome to some of my colleagues, that these biosimilars have the generic name, but with an attached random 4-letter suffix added to the end to distinguish one from the other. We now also have two, so we have filgrastim biosimilars, the short-acting form of G-CSF approved. We also now have 2 pegfilgrastim biosimilars approved, and again, these, to my knowledge, are not yet in the market or in the clinics, but will very shortly be.
Yeah.
And now, as I indicated earlier, we also have an EPO or Retacrit, which has been approved for erythroid stimulation. And now, of course, we're upping the ante for oncologists with the introduction of these cancer treatment biosimilars, trastuzumab and bevacizumab. And just in my own experience in talking with folks, I think concern goes up a bit about efficacy and safety when you're talking about a direct cancer treatment being offered to a patient versus a supportive care agent.
Yeah.
Time will tell if that's the case.
And so, so now we've talked about the definition, the FDA, how they're made, and the ones on the market. Let's just go next to extrapolation and interchangeability. So, as you mentioned earlier, and again, why would you develop a biosimilar if you had to do exactly the same with the brand person, because it'd be prohibitively expensive? So you can take one trial where it works and say, well, if there's 3 other, 4 other, 5 other labeled or compendium indications, I can use it there as well. So that would be extrapolation. But then what about interchangeability? If I go to the drugstore or even in the hospital, I prescribe something that's not in the biosimilar arena, the pharmacist can, with or without my knowledge, change so-called interchangeability to some other drug. This is not yet true in biosimilars?
This is not yet true, although it's there is an allowance for it in the FDA regulations if the manufacturer provides additional interchangeability clinical data to show that changing from the originator to a biosimilar or switching back and forth doesn't have a meaningful impact on efficacy and safety of the biologic, that they can grant a designation of interchangeability. And so an interchangeable biosimilar would be a notch above just being granted a biosimilarity. This has not yet been done in the US, but there are regulations now in place. So if a company wants to go to the expense and they see a business case for doing the additional studies to request from the FDA that designation, they could do such, and maybe it would have some market advantage. We all have patients, that we maybe treat up north, and then they go to Florida for the winter, and maybe the place where they're being treated uses a different agent or a biosimilar. To have that interchangeable designation could be an advantage, perhaps, but that's not yet happened, and it's yet to be seen whether that'll really provide a market advantage to a drug, to a biosimilar going forward.
So now we have these drugs coming to us, whether in treatment or in supportive care, that are rigorously defined and convinced the FDA of their safety, efficacy, and clinically insignificant impurities. We are told, of course, that the promise here is competition and cheaper. So I think, as you and I were emailing back and forth today, it came to our attention just something that Michael Fish published in a letter to the editor in JAMA Oncology, and for our listeners, it's November 1, 2018, where two filgrastims, biosimilar and brand product, not too different pricing. So what are you seeing or hearing in terms of this promise of cheaper and more competition? Is it playing out yet, or will it play out?
Yeah. Well, of course, our hope is it will play out, and it certainly has in Europe, where they found that when not so much a single biosimilar, but if there are several in-class biosimilars on the market, that prices do begin to come down. It doesn't come down the 80% or so you might see with a generic drug, which is much easier to manufacture. For biosimilars, they're seeing maybe a 20-25% drop in price, but these are very expensive drugs, so it may be meaningful.
Very significant.
That may not happen with a single biosimilar, and it's really, at this point, too early to really know. So the research letter that the group, the study that you mentioned recently published, they looked at the experience over the initial couple of years of GCSF biosimilar approval in the U.S. Now they included, and this gets to another complexity here, tibofilgrastim, which in Europe is approved as a biosimilar, but when it was brought to the FDA back 4 years ago, the FDA rules were not in place, so they decided to go for approval as an innovator. So although in this paper in JAMA Oncology, it's listed as a biosimilar, they did all the clinical trials necessary to get approval as an originator. The offshoot of that, of course, is that the Tibo product has not been granted extrapolation. So the first real biosimilar to come along, which is the Sandoz product, filgrastim SNDZ, did get extrapolation. And so if you consider them both as biosimilars, you can do the type of analysis that Mike Fish and his colleagues did, comparing it to filgrastim. But the real comparison here, and it's in the second table in that paper, is between filgrastim, the original GCSF short-acting product, with the first real biosimilar, which is the SNDZ filgrastim. And there we see about a 10-11% decrease in price with the biosimilar product. Now, this is very early. The data is over a year old. And of course, this is something that we're going to have to track over time. And especially as the cancer treatment biosimilars come onto the market, it'll be interesting to see how much real impact there is on the cost of these drugs going forward. As I said, they're very expensive drugs. So even a 10 or 20% decrease in the pricing couldn't be real dollars. But I would just say at this point, this is first look at the very earliest experience here. To me, it's a matter of glass half empty or half full. I see it as half full here because we do see with the biosimilar about a 10% decrease in price in the first couple of years of availability. But again, time will tell. But hopefully, as more and more GCSFs biosimilars get on the market, and the same for these other drugs, the more competition, hopefully the greater the forces, market forces, to lower the price.
Well, I think at 10, 11%, it's already starting to happen. The promise of 30% may have been a bit much to hope for in the beginning, but that's a start.
Yeah.
Well, just a couple more points to cover. I just wanted to ask, because I know my answer, do you feel the biosimilars, if you had to use a therapeutic or a supportive care, they would be just as effective as the brand product? I'm convinced by the rigor of the process that I would use it. Are you doing the same or seeing the same with your colleagues?
Yeah, well, I am. Again, of course, I've been studying this for a long time. You mentioned at the beginning the ASCO task force and the position statement they put out. We looked at the whole analytic process in detail, and were convinced, and this was published in JCO earlier this year as a position
Let me mention
document
that's the April 2018 issue. It's a beautiful article. Gary is first author, and it's called the ASCO Statement Biosimilars in Oncology. So for our listeners.
Yes, and you know, we did a sequel to that. It was in the New England Journal in May, where we went into greater depth. But you can tell from these articles that we really exhaustively looked at the process and feel that it's safe to use these products. I would just add that, of course, as you may know, the biosimilar growth factors, GCSFs, have been integrated into both ASCO and NCCN guidelines as equal options for clinicians planning on supportive care. So patients getting high-risk chemotherapy for neutropenic complications, that the biosimilars or the originator can be equally used, and you can be comfortable, your patient should be comfortable that they will get the same efficacy and safety out of these. Again, as we move into the trastuzumab era and probably next year into rituximab and others, again, it's going to raise the level of discussion, perhaps raise the level of concern, but I am convinced that the process in place and the European experience for over a decade now tells us that this is a very thoughtful, safe process that should improve access to care, hopefully eventually rein in costs, and everybody will benefit, the health system, but individual patients as well.
And I'm looking just for our listeners, that subsequent article you mentioned, New England Journal, is the May 24 issue, 2018, with a nice title, Rationale, Opportunities, and Reality of Biosimilar Medications. Well, I think that really covers our topic. And so unless you had any final comment, Gary, I think we'll wrap up our nice podcast.
Absolutely. No, thanks. Thank you, David.
Well,
it's been great talking with you, and thanks for bringing focus to this topic.
Well, you're very welcome. I think it's a great, fascinating topic, which is in process of evolving.
And that concludes this edition of Blood and Cancer. We will be back with all new interviews and episodes in the 47th episode. For MDedge, I'm Nick Andrews.